Hello.
Hello, can you hear me, sir?
Yeah, ma'am. Please proceed, madam.
Yes, sir. Sir, can you see the screen, sir? Hello?
Yes, madam.
Can you see the screen, sir?
Yeah.
Can I start, sir?
Yeah, please proceed, madam.
Yeah. Hello, everyone. So, welcome to Tauhid Foundation for Allied Healthcare Sciences, Bengaluru. So, today we'll be focusing on carbohydrates. Under carbohydrates, now we'll be focusing on chemistry of carbohydrates. Myself, Rafia, from the Department of Biochemistry. So, coming to introduction, you can see whenever you heard hear the word carbohydrates, whenever when can you hear the word, when have you heard the word carbohydrates? So when it comes to food, so when it comes to food, we'll be speaking about carbohydrates, proteins, lipids and all. So there you have heard the word carbohydrates. So that means carbohydrates, it is related, something related to food or energy. So carbohydrates, it is a macronutrient. Now, the word carbohydrate, it is derived from the general formula CnH2O whole to the power n. So, this is the general formula for carbohydrates that makes them seem to be hydrates of carbon. The term carbohydrates and saccharides, they are closely related. That means carbohydrate, it is related to saccharide. So, saccharide means something which is sweet in taste or sugar. Okay, something which is sweet in taste is known as saccharide. So, the term carbohydrate and saccharide, they are related to each other. That means they are closely related. And the most abundant carbohydrate presents are polysaccharides. So among all the carbohydrates, the most abundant means the most carbohydrates which are present in large quantity are polysaccharides. So poly means many and saccharides, as I said, it means sweet, sweet or sugar. That means polysaccharides means it is a carbohydrate which contains many sugar units. Okay. So most important carbohydrate among all the carbohydrates is glucose. And you know, our body, all the cells of our body, they are completely dependent, they are dependent on energy for, they are dependent on glucose for energy. So the word saccharide, it has come from it comes from the word table sugar or sweet. And in several languages, like in Sanskrit, it is known as sarkara. In Greek, it is known as saccharum, and in Latin, it is known as sacharum. So these are the different words which is resembling saccharide word. Okay. So next, coming to what is the definition of carbohydrates. So carbohydrates may be defined as polyhydroxyaldehyde or polyhydroxyketone or compound that yields these derivatives or hydrolysis. So like this, you can see when we say aldehyde, it will be coming like CHO will be there. Okay. And when we say ketone, you can see C double bond O. So this is a structure of glucose and this is a structure of fructose here, whichever you are seeing here. Okay. Now next, coming to what is the importance of carbohydrates. So carbohydrates, they are distributed widely in nature. As you know, we are complete, our body cells, they are dependent on carbohydrates for energy. They are the key intermediates of metabolism. So on metabolism, they'll be producing sugars. They are the structural components of plants. That means in plants, they are present in the form of cellulose. They are central to materials of industrial products like paper and fibers. And lastly, they are the key component of food sources like sugars, flour and vegetable fiber. Next, coming to now, till now we were studying what are carbohydrates and what is the importance of carbohydrates. Now coming to what are the functions of carbohydrates. Actually, there are enormous functions of carbohydrates. Carbohydrates plays a very important role in our body. Among them, we will be studying few functions. Like first, Carbohydrate, they are the source of energy in our body. So they are the most important source of energy in our body. So from carbohydrates, I said, which is the most common carbohydrate present in our body is glucose. So as I said, our body cells, they are dependent on glucose for energy. So it is one of the function of carbohydrate. Then they are carbohydrates, they are the intermediates of metabolism. So on metabolism, they'll be producing energy. Then they are the storage form of energy in our body. So this glucose, whatever glucose which is produced from carbohydrates, it is converted to glycogen and it is stored in our body for further metabolism, for further energy purpose. Okay. Then carbohydrates, they are the structural components in our body. They also act as fibers. They are the constituents of nucleotides. And finally, they help in detoxification in our body. Next, coming to what are the classification and nomenclature of carbohydrates. So in this class, we will be studying what is the basic of carbohydrates, means like what are carbohydrates, how they are formed, how they are classified, what are their nomenclature. So we'll be focusing on that. Further, in the later classes, we will be focusing what is the metabolism of carbohydrates. Okay. So now coming to what is the classification of carbohydrates. So usually carbohydrates, they are broadly classified into 3 groups, or further like it can be classified into 4 groups also. Under oligosaccharides only disaccharides will be coming. Sometimes you can take disaccharides as a different group, but usually they are classified into 3 different groups, that is monosaccharides, oligosaccharides and polysaccharides. So coming to monosaccharides, this mono means something which is single. So monosaccharides, they are the simplest form of carbohydrates which cannot be further hydrolyzed because it is only one. See, if you are having one chocolate, you cannot break it further. So if you are having 2 chocolates, it is like oligo or di, we can break it to 1 plus 1. But if it is mono, that means you are having, it is like single. So monosaccharides, they are the carbohydrates which are made up of single sugar unit. Then coming to oligosaccharides, they are made up of 2 to 10 sugar units, and polysaccharides, poly means many. So they are made up of more than 10 sugar units. These sugar units, either it can be same type of sugar units or sometimes it may be different types of sugar units. Okay. So next coming to, in detail, we'll be studying what are monosaccharides, oligosaccharides and polysaccharides. So these monosaccharides, again they are further divided based on their functional group and based on the number of carbon atoms. So based on the functional group, they are further divided as aldoses and ketoses. So under aldoses, example is glucose. Under ketoses, example is fructose. Okay. So based on this monosaccharides, based on number of carbon atoms, again they are further divided as trioses, tetroses, pentoses, hexoses and heptoses. Then next coming to oligosaccharides. As I said, oligosaccharides, these are the carbohydrates which will be having 2 to 10 monosaccharide units. That means they'll be having 2 to 10 sugar units. So again under disaccharides, we are oligosaccharides, we are having disaccharides, trisaccharides and tetrasaccharides. So under disaccharides, we are having examples are maltose, lactose and sucrose. So maltose, this is a sugar which is present in your malt. For example, your ragi malt and all, which we prepare to feed the babies like infants and all, no, that in that it is present. So that sugar is known as maltose. Lactose, it is a sugar, it is also known as milk sugar. This is a sugar which is present in milk. And sucrose, it is a common sugar or table sugar which we use in our daily like home routine and all. So that type of sugar is known as sucrose. So these are the examples for disaccharides. Coming to trisaccharides, under trisaccharides we will be having raffinose. Under tetrasaccharides we will be having stachyose. So next coming to polysaccharides. As I said, poly means many. So poly means many and saccharide means sugar units. So polysaccharides, these are the carbohydrates which are having many sugar units, means they'll be containing more than 10 sugar units. So again under polysaccharides, we are having 2 types, that is homopolysaccharide and heteropolysaccharide. So homo means same. So as I said, polysaccharide means they'll be having more than 10 sugar units. So that means if they are having more than 10 sugar units and if the sugar units are same, if it repeatedly the same sugar units are there, for example, like glycogen. So glycogen is made up of only glucose. So like I think like this glycogen is having 50 glucose, means repeatedly 50 glucose are combining together to form one polysaccharide. Those type of polysaccharides are known as homopolysaccharides. Heteropolysaccharides means these are the polysaccharides which are having different monos, like sugar units. Either it can be glucose, either it can be fructose or galactose. So different sugar units combining together to form a carbohydrate. Such carbohydrate is known as heteropolysaccharide. So under homopolysaccharide, we are having starch, dextrin, glycogen, cellulose, and inulin. Under heteropolysaccharides, we are having hyaluronic acid, heparin, chondroitin sulfate, dermatan sulfate, and keratin sulfate. So now in detail we will be studying single carbohydrate units. Okay. So first coming to monosaccharide. So example for monosaccharide is glucose. So as I said, mono means one and saccharide means sugar. So monosaccharides, these are the carbohydrates which are made up of single sugar unit or one sugar unit. Since it is having one sugar, it cannot be further broken down or hydrolyzed. So monosaccharides, again, since they cannot be further hydrolyzed, but still they can be further divided. So monosaccharides, can be subdivided into different groups based on function and based on number of carbon atom, as we have discussed earlier. So based on functional group, based on their function, they can be further divided as aldoses or ketoses. Aldoses means these are the carbohydrates which is having CHO in their structure. Or ketosis means these are the carbohydrates which are having C double bond O in their structure. So these monosaccharides, again further they can be divided depending upon the number of carbon atoms they are having in their structure. So if they are having 3 carbon atoms, they come under trioses. If they are having 4 carbon atoms, they come under tetroses. If they are having 5 carbon atoms, they come under pentoses. And if they are having 6 carbon atoms, they will be coming under hexoses. And 7 carbon atoms, they will be coming under heptoses. So now we will be studying what is the classification based on the number of carbon and what are the aldoses and ketoses coming under this. So if the number of carbon atoms are 3, so there will be the type of sugar is trioses. Aldoses coming under trioses are glyceraldehydes. Okay, example. So ketoses coming under trioses are dihydroxyacetone. So if the carbohydrate containing, if the monosaccharide contain 4 carbon atoms, then they will be coming under tetroses, and the aldoses coming under tetroses is erythrose, and the ketoses coming under tetroses is erythrulose. So you need to remember these examples. Like for example, under trioses, the aldoses are glyceraldehyde. Under trioses, the ketoses are dihydroxyacetone. So if the monosaccharide contains 5 carbon atom, so the type of sugar is pentoses. So under pentoses, the aldoses are ribose and xylose. And under pentoses, the ketoses are ribulose and xylulose. The next coming to, if the monosaccharide contains 6 carbon atom in the structure, then the type of sugar is hexoses. So under hexoses, the aldoses are glucose and galactose, and the ketoses are fructose. That means this glucose, it is having CHO in their structure. Since aldoses, here as I said, you can see aldoses, they'll be having CHO, and ketoses will be having C double bond O. Okay, so the hexoses, under hexoses, the ketoses are fructose. Then next coming to heptoses. So if the number of carbon atoms are 7, the type of sugar is heptose. The aldoses coming under heptoses are glucoheptose, and the ketoses is pseudoheptulose. Okay. Then next coming to, what are the examples and importance of these sugar units. Okay. So as we saw, under trioses, we are having glyceraldehyde and dihydroxyacetone. So the importance of these trioses is they are the intermediates of glycolysis. So glycolysis, this will be coming under metabolism of carbohydrates. So this is a pathway where glyco means glucose, lysis means breakdown. So we are breaking down glucose to give energy. So that cycle is known as glycolysis cycle. So under glycolysis, you will be getting all these trioses. Okay. Then they are the precursor for glycerol formation. Okay. Then Next, coming to tetrosis. So under tetrosis, de-erythros will be coming. The importance of erythros is they are the intermediates of again carbohydrate metabolism. So whenever we are metabolizing carbohydrates, we will be getting erythros. The next, coming to under pentosis, we are having ribose. So the importance of ribose is they are the structural element of nucleic acid, RNA and coenzymes. That means they are the structural element of nucleic acids like DNA, and they are also structural elements of RNA and formation of many coenzymes. Okay. Then next, coming to hexoses. So hexoses, they are very important. So under hexoses, we are having glucose, fructose, galactose and mannose. So coming to what is the importance of glucose in our body? They are the main sugar of the body. That means they are the main sugar unit on which our body cells are dependent for energy. Coming to fructose, so whenever our body we are having something and we will get polysaccharides. So from polysaccharides we'll be getting glucose, fructose and all these monosaccharides. So whenever the glucose has become excess, then it is converted to fructose. So this fructose, whenever it is there in our body, it will be stored in liver. So whenever our body is in need of glucose, that time what will happen? This fructose, it is converted to glucose and then it is utilized by our body. Okay. The next, coming to galactose, it is synthesized in mammary gland to make the lactose of milk. So this lactose, as I said, lactose, it is known as milk sugar because it is present in milk. This lactose, it is a disaccharide. So this disaccharide means it is made up of two monosaccharide units. So these two monosaccharides, what are those? These are galactose and glucose. So this galactose, it is helping in formation of lactose, that is milk sugar. Then next, coming to mannose. Mannose, it is a constituent of glycoprotein or glycolipids. Okay. So we'll be moving further. So this was about monosaccharides. So we studied what are monosaccharides. What is the classification of monosaccharides? So they are divided into two groups depending upon the function and depending upon the number of carbon atoms they'll be having and what is their importance. Now coming to disaccharides. So see, you can see this is one monosaccharide and this is one more monosaccharide. So two monosaccharides combining together to form one disaccharide. So they are combining together. That means they are holding each other hand to become one. No, two things are standing here and they are holding together to form one. So this is a disaccharide. So the bond present between them, between one monosaccharide and another monosaccharide, this bond is known as glycosidic bond. Okay. So this bond, this is glycosidic bond, which is very, very important in combining two monosaccharide units. Okay. So under disaccharides, lactose, maltose and sucrose, these are the examples for disaccharides. So di means two and saccharides means sugar units. So disaccharides they'll be made up of two monosaccharides or two sugar units. So disaccharides, they consist of two monosaccharide units held by glycosidic bond. So as I said, see here there are this is one monosaccharide unit and this is one more monosaccharide unit which is combined together and they are held together by one bond. This bond is known as glycosidic bond. They are crystalline, they are water soluble and sweet in taste. Okay, because like we can see even milk is having some amount of sweetness in it, and even malt also will be having some amount of sweetness, and usually sugar is known for sweetness only. So they are crystalline disaccharides, they are water soluble and they are sweet in taste. They subdivide based on the presence or absence of free reducing group. Again, these disaccharides, they are further divided as reducing disaccharides and non-reducing disaccharides. So what are reducing disaccharides? So reducing disaccharides, they'll be having free aldehyde or ketone group. So these reducing disaccharides, they'll be having free aldehyde or ketone group. Examples for reducing disaccharides are maltose and lactose. Means it is the sugar present in malt and sugar present in milk. So non-reducing disaccharides. So these what are non-reducing disaccharides? They do not have any free aldehyde or ketone group. So example for non-reducing disaccharide is sucrose or common sugar which we use in our daily routine. Okay. So next, coming to oligosaccharides. So oligosaccharides, as I said, they'll be having 2 to 10 monosaccharide units. So 2 to 10 monosaccharides units combining together joined by glycosidic bonds comes under oligosaccharides. So they are joined together by a specific bond called glycosidic bond. So on hydrolysis, they give rise to 2 to 10 molecules of simple sugar units. So when we break down oligosaccharides, we will be getting 2 to 10 single sugar units. Okay. Then they are subdivided based on the number of monosaccharide units. So based on how many monosaccharide units they are having, they are further subdivided. If they are having 2 monosaccharide units, then it is known as disaccharides. Means di means 2. Tri means 3. So if they are having 3 monosaccharide units, they'll be coming under trisaccharides. If they are having 4, they'll be coming under tetrasaccharide. If they are having 5 sugar units, they'll be coming under pentasaccharide. Next, coming to classification of oligosaccharides. So, as I said, these oligosaccharides, they are divided based on the number of sugar units present. So if they are having 2 sugar units, disaccharides. So example for disaccharides is maltose, lactose and sucrose. So I said maltose, it is a sugar which is present in malt. Okay, your ragi malt or whatever malt it is. So this maltose, it is made up of 2 glucose unit. Means this is one monosaccharide and one more monosaccharide. So 2 monosaccharide combining together to give disaccharide. So this maltose is made up of glucose and glucose. Lactose is made up of one molecule of glucose along with one more molecule of galactose. And sucrose, which the common sugar which we use, it is made up of one molecule of glucose and one molecule of fructose. So this fructose, it is there, no, this is a monosaccharide. This is sweet in taste. So because of this fructose only, the sugar which we use is a sweetener. No, the sugar which we use, like whenever we have sugar, we get the sweet taste because of this fructose only. Okay, because fructose, it is very sweet in taste. This fructose, usually it is present in honey and some fruits. Okay, then next coming to trisaccharides. So under trisaccharides, I said it is having 3 monosaccharide units. So example is raffinose. So this raffinose, it is made up of one molecule of glucose, one molecule of fructose and one molecule of galactose. 1, 2, 3. That means it is having 3 monosaccharide units. So raffinose, it is made up of glucose, fructose and galactose. Next coming to tetrasaccharides. So if they are having 4 monosaccharide units, so they will be coming under tetrasaccharides. Example is stachyose. This stachyose is made up of 2 molecules of galactose along with 1 molecule of glucose and 1 molecule of fructose. That means it is made up of 1 galactose, 1 more galactose along with 1 glucose and 1 fructose. Okay. So for example, you can take this only. For raffinose, if you add 1 more galactose, it is stachyose. Okay. The next coming to pentasaccharides. So pentasaccharides, they'll be having 5 monosaccharide units. Example is verbascose. So verbascose is made up of 3 molecules of galactose along with 1 molecule of glucose and 1 molecule of fructose. So this is the classification of oligosaccharides. I think now you have got an idea what are oligosaccharides and how they are made up of. So based on the number of monosaccharide, this monosaccharide, it can be either glucose or galactose or fructose. So based on this, how many monosaccharide units are there? Based on that, you will be getting different oligosaccharides like disaccharides, trisaccharides, tetrasaccharides and pentasaccharides. Okay. I hope you have understood about oligosaccharides. Next coming to polysaccharides. So, as I said, poly means many sugar units. So, polysaccharide means it is a carbohydrate which is made up of many sugar units. Here you can see 1, 2, 3, 4, 5, 6, 7. It is like n number. Okay, it is more than 10 sugar units. So, polysaccharide still 10. It is under oligosaccharides. So, if the sugar units are more than 10, if the carbohydrate has more than 10 monosaccharides or more than 10 sugar units, it will be coming under polysaccharide. And the bond present here is glycosidic bond. The bond present between one monosaccharide of to another monosaccharide is glycosidic bond. Okay. So, as I said, polysaccharides, they contain many sugar units, that is more than 10. If they are 2 to 10, they'll be coming under oligosaccharides. If it is 1, it will be coming under monosaccharide. Okay. They have high molecular weight and are sparingly soluble in cold water. So polysaccharides, they are having high molecular weight. Why they'll be having high molecular weight? Because they are made up of more than 10 sugar units. Since they are made up of more than 10 sugar units, they'll be having high molecular weight. They are also known as glycans. Polysaccharides, the other name for polysaccharides is glycans. Okay. They may be either linear or branched in structure. So as we saw here, this is a linear polysaccharide, means they are having single line. See, it is like single line. Either it can be linear or sometimes it can be branched in structure also. Branch means you can remember a tree. Like if there is one tree, it is having many branches among which there are many leaves. So these leaves, they are monosaccharides. So all the leaves combine together to form one tree. So that tree is a polysaccharide. Okay. They are not sweetest and do not exhibit any of the properties of aldehyde or ketone group. So whatever the properties of aldehyde and ketone group are there, this do not exhibit that. Okay. The next coming to, as I said, polysaccharides, they are again further divided into homo and heteropolysaccharide. Homo means same. So if the polysaccharide means what? Many sugar units. That means same many sugar units. So if the polysaccharide, if the carbohydrate contains same amount of sugar, many sugar units, then they are known as homopolysaccharide. Hetero means different. So if a carbohydrate contains different many sugar units, then it is known as heteropolysaccharide. Okay. So example for homopolysaccharides are starch, glycogen and dextrins. Next coming to example for heteropolysaccharides are agar, pectins and glycosaccharides. Okay. The next coming to what is the structural functional relationship of monosaccharides. So till here we studied about polysaccharides. Now I think you might have got an idea what are monosaccharides, what are disaccharides, what are oligosaccharides and what are polysaccharides. So monosaccharides, they are made up of single sugar unit. Disaccharides, they are made up of 2 sugar units. Oligosaccharides, they'll be having more than 2 to 10 sugar units. Then coming to polysaccharides, polysaccharides will be having more than 10 sugar units. Okay. So next we will be moving on to structural functional relationship of monosaccharide. So this is the structure of glucose. Now we will be studying what are the structure of monosaccharides, what are the physical properties and chemical properties of monosaccharides. So coming to structure of monosaccharide. So under monosaccharide, as I said, the important carbohydrate present in our body or the important monosaccharide which we are dependent is glucose. So glucose is the most important physiological and biomedical monosaccharide because our body, it is dependent on glucose for energy. So we will be getting galactose also and even fructose also. But what we studied, fructose, again it is converted to glucose for giving energy. So glucose is the most important physiological and biomedical monosaccharide. It can be represented in straight chain structure, ring or cyclic structure, boat and chair form structure. Okay. So first coming to straight chain structure. So this is a structure of glucose. Since it is having a straight chain, so it is known as straight chain structure. So the molecular formula for glucose is C6H12 and O6. Okay. So you can see here 1, 2, 3, 4, 5, 6. So it is having 6 carbon atoms. Coming to O, it is having 1, 2, 3, 4, 5 and 6. It is having C6O6 and H. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So it is having C6 carbon atoms, 12 hydrogen atoms and 6 oxygen atoms. So this is a straight chain structure of glucose. Next coming to, this is a ring structure of glucose. Okay. Ring or cyclic structure. Okay. So here ring or cyclic structure, what we'll be studying is, for example, this is a ring. Okay. Ring or cyclic structure. Here we are having aldehyde. Along with aldehyde, if we add alcohol, it is forming a a structure. This structure is known as cyclic structure because, or ring structure here, because this C carbon atom, it is attached with 4 different atoms. So this structure is known as cyclic structure or ring structure. Okay. Here we are taking aldehyde. So here you can see this, whichever it is there in black color. Here you can see CHOR. Okay. Now this double bond O, along with double bond O, we are adding H. So it has become single bond, and this O is going here along with this R. So this is known as hemiacetal structure or cyclic or ring structure. Okay. It is also known as Haworth projection. Okay. Next, coming to when we are taking ketone and we are adding with alcohol. Okay. So this ketone plus alcohol, it is giving hemiketal. Okay. Here you can see this C double bond O. Double bond is converted to single bond because H is coming and attaching here. So this O is converted to OH, R, here R and R dash. Okay. So this is hemiketal. So here you can see there are 2 arrows. That means from this structure can be formed. Again, from hemiacetal, we can get back to aldehyde and alcohol. Okay. So it is a reversible reaction. That means from this you can get this also. Again, from hemiacetal structure you can get aldehyde and alcohol also. So it is a reversible reaction. If it was single arrow, that means it from here we can get this, but from this we cannot get this. That means it is a irreversible reaction. Okay. Next, coming to Haworth projection of formula of glucose. So here you can see this is a single line structure. Okay. So here this is known as D-glucose, and here it is a penultimate carbon. Penultimate carbon means just below the last, like before the last carbon atom, whichever atom is there, that is known as penultimate carbon atom. Okay. So here 1, 2, 3, 4, 5. So on the 5th carbon atom, we are having OH group. Okay. So what is happening? This OH, it is going here and combining this H, it is going from here to here. So here you can see double bond is converted to single bond, and this O is forming a ring structure. So this is known as alpha D glucuronase or Fischer structure of glucose. Okay. So along whenever this we are adding pyranose. So for this, if we add pyranose, we will be getting this structure, this ring structure. Okay. So this is known as alpha D glucuronase or Haworth structure of glucose. Okay. So this is the Haworth projection of formula of glucose. So from single line structure, okay. So we can get it into alpha D glucuronase, that is Fischer structure. From that we can get Haworth structure of glucose. Then next, coming to, this is same as it is, like this is a Haworth structure of fructose. Here what is happening? Same thing is happening here. This H we are sending, this OH we are sending here. So here you can see this OH is attached to this, and we are getting here O. Okay. So this is the alpha D fructofuranase. From this, if we add furanose, we will be getting alpha D fructofuranose. So this is a ring structure or Haworth structure of fructose. Okay. Then next, coming to what are the properties of monosaccharides. So this is very, very important. So this is the chemical, now we will be studying the chemistry. Now exact chemistry of carbohydrates we will be studying. Okay. So under this, first coming to what are the properties of monosaccharides. So here under this we will be studying isomerism and what are the chemical reactions it will undergo. Okay. So coming to isomerism. The compounds possessing same molecular formula and different structures are referred to as isomers. You know what are isomers? So isomers, these are the substances or compounds which will be having same molecular formula but different structure. So the molecular formula will be same, but it will be having different structure. So those type of compounds are known as isomers. Okay. So the phenomenon of existence of isomer. So this phenomenon which is happening is known as isomerism. Okay. So under isomerism, we will be studying 2 types, that is structural isomerism and stereoisomerism. Okay. In detail, we will be studying single isomerism. So coming to structural isomerism. So they are having same molecular formula but differ in their structure. So as we said, it is the word itself, it is saying isomerism means they are having same molecular formula but different structure. So under structural isomerism, they are having same molecular formula but they are having different structures. For example, aldose, ketose, isomerism. So glucose and fructose, they are isomers of each other because glucose, the molecular formula for glucose is C6H12O6. Even the molecular formula for fructose also, it is C6H12O6, but they are having different structure. The molecular formula is same for glucose and fructose, but the structure is different. These type of isomers are known as structural isomerism. Okay, they are having same molecular formula, but they are having different isomers of each other. Okay, but differ in their structural formula. Okay, molecular formula will be same, but structure will be different. That is, structural formula will be different. Okay, so here you can see this is having C6, 1, 2, 3, 4. Now we are studying what is aldose ketose isomerism. So this is the structure of glucose and this is the structure of fructose. Okay. Now glucose is having 1, 2, 3, 4, 5, 6. That means it is having 6 carbon, 12 hydrogen atoms. That is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 and 12. That means it is having 12 hydrogen atoms and 6 oxygen atoms. Here you can see 1, 2, 3, 4, 5 and 6. So it is having C6H12O6. So this is the what it is the molecular formula of glucose. So this is the molecular formula of glucose and this is the structural formula of glucose. Okay, but the coming to molecular formula of fructose is it is having 1, 2, 3, 4, 5, 6. It is having 6 carbon atoms and 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 and 12. That is, it is having 12 hydrogen atoms and 6 oxygen atoms. 1, 2, 3, 4, 5, 6. So it is having 6 oxygen atoms. That means the molecular formula for glucose also it is C6H12O6 only, and the molecular formula for fructose also it is C6H12O6 only. What is the difference here? The difference is structure. You can see this will look different from this. No, this is having a different structure and this is having both are D-glucose and D-fructose only, but still it is having different structures. The molecular formula is same, but the structure is different. These are the examples for isomers and these are the examples for structural isomerism. Okay, then next coming to stereoisomerism. So here the molecular formula is same, the same molecular formula, structures but differ in configuration. Okay, so it is having same molecular formula and same structure, but different configuration will be there. Okay, so asymmetric, that is carbon allow the formation of say stereoisomerism. So there is something known as asymmetric carbon atom. Okay, so this asymmetric carbon atom which is there. So asymmetric carbon atom means it is attached to 4 different, like for example, now this carbon, it is attached to 4 different atoms. So this is known as asymmetric carbon. So this asymmetric carbon allow the formation of stereoisomerism. So in structural isomerism, we saw they are having same molecular formula but different structure. In stereoisomerism, they are having same molecular formula and even structure also, but they are different in their structural configuration. Okay. So next coming to what are the types of stereoisomerism of glucose. So we are having D and L isomerism, optical isomerism, epimerism and anomerism. Okay, so these are the types of stereoisomerism of glucose, and here you can see we are focusing on glucose because glucose is the only carbohydrate on which our body is completely dependent. So glucose, carbohydrates, they are the basic source of energy for our body. So among carbohydrates, glucose is the most abundant carbohydrate which is present in our body. So we are focusing on glucose. Okay, so next coming to what are the D and L isomerism, that is enantiomer. So here you can see D-glyceraldehyde. In D-glyceraldehyde, the OH is on the right side. Okay, but in L-glyceraldehyde, the OH is on the left side. The full structure it is same. So it is having structure also same and even molecular formula also it is same, but it is different in their enantiomer. Here you can see in D-glucose it is on the right side and in L-glucose it is on the left side. So these are known as enantiomers. Okay. So in structural isomerism, we saw glucose was compared with fructose, but in enantiomers, Here we are comparing glucose with glucose only. Okay. So here you can see, like it is also known as mirror image. So whenever you see something under mirror, have you observed whatever is there in the right side, it will be appearing on the left side, and whatever is there in the left side, it will be appearing on the right side in the mirror whenever we see. No. So D and L isomers are mirror images of each other. So whatever is there, see here you can see whatever is there on the right side, here it is appearing on the left side. And whatever is there is on the left side, it is appearing on the right side. So we can say in enantiomers, this D glucose and L glucose, they are the mirror images of each other. So these two together, it will form enantiomer. Okay. So next coming to optical isomerism. So optical activity is the capacity of a substance to rotate the plane polarized light passing through it. So here you can see it is here we are getting light. We are focusing on light. Okay. So optical activity, it is a capacity of a substance to rotate the plane polarized light passing through it. So whatever light is passing through it, it is having the capacity to rotate. So that activity is known as optical activity. So when the light rotate to right, that is clockwise direction, that substances is said to be dextrorotatory, or you can write down D. So here we are using in enantiomers, we are using capital D and capital L. So here in optical isomerism, you are not supposed to use capital D. So only you are supposed to use small d, or you can write it as plus. Okay. But when the light rotate to left, that is left direction, that is anti-clockwise direction, those substances can be said as levorotatory, or you can use small l or minus. Okay. So when equal amount of D and L isomers are present, that mixture is known as racemic or DL mixture. So here we are seeing what is the capacity of a substance to rotate light. Okay, so whenever you are having something and through that you are passing the light, if the light is passing in the clockwise direction, means it is passing in the right side, that type of light is known as dextrorotatory, those type of substances activity. Okay, so whenever we are passing the light, and if it is passing in the opposite direction, that is in the anti-clockwise, those type are known as levorotatory. Okay, so if the mixture, it is having same amount of D, that is dextrorotatory and levorotatory, the isomers are present, those type of mixture is known as racemic mixture or DL mixture. Okay. Next coming to epimerism. So epimers are the sugars which are differ with each other with respect to single carbon other than anomeric carbon. So here we will be having single carbon atom will be different. Okay, for example, galactose and mannose, they are the epimers of glucose. That means glucose, galactose, mannose, they are having same structure, but they'll be having one difference, that is they'll be having in single carbon atom difference. Okay, they differ from glucose with respect to C4 and C2. So in the glucose structure only, in the fourth carbon atom, there'll be some difference. So that is known as galactose. So in the glucose structure only, there'll be some difference in the second carbon atom. So that comes out, that is the structure of mannose. So these type of sugars are known as epimers, and this reaction is known as epimerism. Okay, so here you can see example I am showing. See, this is the structure of glucose, which we are very familiar with the structure of glucose. So it is having C6H12O6. Okay, so galactose also is having C6H12O6 only, and mannose also. But what is the difference means, if they take the structure of glucose, here in the fourth carbon atom, you can see here in the glucose, in the left side we are having H, and in the right side we are having OH, that is hydroxyl group. But in the galactose, we can see in the fourth carbon atom, the hydroxyl group has shifted to left and the hydrogen group has shifted to right. The structure is same only there is difference in the fourth carbon atom. There is exchange between hydrogen and hydroxyl group. So this is the structure of galactose. Okay, then in mannose, you can see, you can compare this structure with this. Okay, here this structure is same as this only, but in the second carbon atom, you can see what is happening. The hydroxyl group is right in the glucose and hydrogen group is on the left. But in mannose, you can see the hydroxyl group has shifted to left and the hydrogen group has shifted to right. Okay, so you can see, you can say galactose and mannose, they are the epimers of glucose, means they are having structure similar to glucose only, but there is one difference in the one carbon atom. Sir, what is the definition of epimers? They are having epimers are sugars which are differ with each other with only respect to one single carbon. Only in the one carbon atom there will be changes. As you can see, in galactose there is changes in the fourth carbon, and in mannose there is changes in the second carbon. But the structure is similar. It assembles glucose only, no? So these are the you can say galactose and mannose. They are the epimers of glucose. Okay. Next, coming to an anomerism. Okay. So in solution, glucose predominantly exists as closed chain structure because of cyclization of sugar. An addition asymmetric center is created at C1, that is anomeric carbon. This leads to formation of two isomers known as alpha D glucopyranose and beta D glucopyranose. This thing is known as anomerism. Okay. So whenever we dissolve glucose in water, okay, in some solution. So in that solution, what will happen? This glucose, it will be existing as closed chain structure. Okay. Then later on, because of cyclization of sugar, an additional asymmetric center is created at first carbon atom. Okay. That is anomeric carbon. So this structure, it leads to formation of two isomers, namely alpha D glucopyranose and beta D glucopyranose. Okay. So here you can see, this is the structure of glucose. Here what is happening? The hydroxyl group is on the right side, and the hydrogen group is on the left side. Okay. Usually here it will be C double bond O. Okay. So, but what is happening? Whenever we are dissolving it in a solution, it is happening alpha D glucose. Here in alpha D glucose, we can see hydroxyl is on the right side and hydrogen is on the left side. But in the beta D glucose, we can see hydroxyl is on the left side and hydrogen is on the right side. So these are the anomers of glucose, and this reaction is known as anomerism. Okay. The next, coming to mutarotation. Okay. In mutarotation, it is the word itself, it is saying mutarotation. So it is defined as the change in specific optical rotation representing interconversion of alpha and beta forms of D-glucose to an equilibrium mixture. So what is happening here? Here you can see we are having one equilibrium mixture. Okay. And here the specific rotation is at plus 52.7 degree. Okay. So, but from alpha D, this equilibrium mixture is forming, and from beta D also we are getting equilibrium mixture at specific rotation. So this is known as mutarotation. Here you can see this is a structure of glucose. Okay. So under at plus 52.7 degree, but from D we are getting alpha D. In alpha D, what is happening? You can see here this OH is going and combining here, and this O with this H, no, this O, with this O, this H will go and combine here. And this O, which is there, it is in the ring form. So this is the alpha D structure. Okay. Here you can see in beta D, the hydroxyl is on the left side and the hydrogen group is on the right side. And here this is a reversible reaction. So here you can see two arrows. That means from D glucose we can get alpha D glucose. Again from alpha D glucose we can get D glucose. From D glucose we can get beta D glucose. From beta D glucose again we can come back to D glucose. So this is known as mutarotation. Okay. So these are the properties. Now we studied these are the properties of monosaccharides. So under properties of monosaccharides we studied isomerism and chemical reactions. So we will just have a brush up of what are the properties of monosaccharides. Under isomerism, as I said, they are having same molecular formula but different structure. Okay, they'll be having same molecular formula. Example for isomers are glucose and fructose. Glucose and fructose, the molecular formula for glucose and fructose is C6H12 and O6. But they are having different structure. Those are known as isomers. Okay. Under isomers, again we are having structural and stereoisomerism. So structural isomerism means they'll be having same molecular formula but different structure. Here you can see the molecular formula for glucose is this, and for fructose it is this, but it is having different structure. So these are the example for structural isomerism. Next, coming to stereoisomerism, they are having same molecular formula, same structure but different configuration. Okay, so this is the example for stereoisomerism. So here you can see D and L. As I said, they are the mirror images of each other. So they are having same structure and same molecular formula, but they are having different configuration. The configuration is different here. So they are known as, and here what is happening, whatever is there on the left side, it is coming on the right side, and whatever is there on the right side, it is coming on the left side. So these are known as D and L structure of a glucose. Okay. The next coming to optical isomerism. So as I said, if the light is passing through a substance, if the light, it is rotating through the right, that is clockwise direction in that substance, then it is known as dextrorotatory. But when the light, when a capacity of light, or when plane polarized light is passing through a substance, and if it is moving in the left side or anti-clockwise direction, then it is known as levorotatory. And if a substance, if a solution is having, or if the mixture is having equal amount of D and L, that is equal amount of dextrorotatory and levorotatory, those type of mixture is said to be racemic or DL mixture. The next coming to epimerism. Epimerism, these are the sugars which are different with each other with respect to single carbon. So as I said, epimers of glucose are galactose and mannose. So galactose and mannose, they are having structure similar to, molecular formula similar to glucose, but there is different in single carbon. So in galactose, there is difference in the fourth carbon, and in mannose, there is difference in the second carbon atom. You can see here. No. So these are the epimers of glucose. Next coming to anomerism. So in anomerism, in a solution, the glucose will be existing in the closed state, and here you can see it will be further divided as alpha D glucose and beta D glucose. So because of cyclization. Okay. So an additional symmetric center is created. So this is known as anomeric carbon, because of which leads to the formation of two isomers, namely known as alpha D glucopyranose and beta D glucopyranose. Okay. Next coming to mutarotation. So in mutarotation, you can see here from D glucose we are getting alpha D glucose and beta D glucose at this degree plus 18.7 degree. Okay. So this is mutarotation. So I hope you have understood the properties of carbohydrates. In the next class, we will be continuing with the part 2, that is chemistry of carbohydrates, and we will be continuing with the chemical properties of carbohydrates. Okay.
Thank you, madam. Thank you so much.
Okay, sir. Thank
You
you, sir.
explained very well, madam. Yeah. A detailed explanation regarding carbohydrates.
Yes, sir.
And
Sir,
even a DMLT. Yeah.
if
Tell
they
me.
understand this, no, sir, for them it will be very easy to understand the cycle, sir. Once they have understood the chemistry of carbohydrates, cycles will be very easy for them. Metabolism will become very easy for them.
Yes, madam.
Okay, sir.
So
Thank you, sir.
I think even DMLT students, they can understand what you have taught today.
Yes, sir.
So
Sir, yes, sir. This is there for diploma also, sir. These are the basic things of carbohydrates.
That's right. That's what I'm telling. That's what. So very nice class, madam. Thank you, madam. Thank you so much.
Okay.
So tomorrow we will continue with this.
Okay, sir. Okay. Thank you,
Thank
sir.
you.
Thank you so much. Okay,
Shall I wind up, madam?
yes, sir. Sure, sir.
Yeah.
Okay.
Thank you.
Thank you, sir.